Hello, I'm Dr. Elana Miller. Today we're going to deep dive into a study published in the Journal of Geroscience titled Topical Rapamycin Reduces Markers of Senescence and Aging in Human Skin, an exploratory prospective randomized trial. We'll be defining what we mean by the science of aging, examining the methods of the study, reviewing the results, and discussing the implications and insights as it relates to aging and the biological processes that drive aging. If you're familiar with mTOR-driven aging, feel free to skip to the timestamp for the study, but if not, please stick around to learn about mTOR, cellular senescence, and the molecule rapamycin. First, it's important to understand the science of growing old. You don't just get wrinkly and die for no reason. Aging is driven by multiple biological factors, but today we're focusing on processes related to mTOR-driven aging. mTOR is a protein kinase. It regulates functions of the cells such as growth, survival, and metabolism, and more. This protein kinase activates and deactivates as it fulfills its biological duties, but when it's activated for too long, side effects include out-of-control cell growth and the accumulation of damaged or mutated cells. As we get older, mTOR may even stay active all the time. These dysfunctional cells are usually recycled through a process called autophagy, but some of these cells slip through the cracks and become senescent cells. In any discussion about aging, we need to define cellular senescence. As you know, our entire body is made up of cells that work hard to keep us healthy and running. They divide and make copies of themselves to propagate new cells, but when a cell becomes dysfunctional, it is broken down and recycled so as not to damage our body. A senescent cell is one that has become dysfunctional, escapes recycling, and no longer divides. Not only do these cells simply persist in the body doing a poor job of whatever they were before, be that blood cell, tissue, brain cells, etc., but they also secrete pro-inflammatory molecules that can cause damage to surrounding tissues and push other cells to become senescent. As we age, we gain a higher number of senescent cells thanks to factors like overactive mTOR. This means our bodies become more and more run by these dysfunctional cells rather than new, healthy ones. Functions degrade, tissue breaks down, and many adverse conditions and diseases arise. This degradation happens throughout the body, but today we're looking through a trial made to slow down mTOR-driven aging in skin tissue through the use of topical rapamycin. Rapamycin, a molecule known for its ability to inhibit mTOR activity, has drawn significant interest from researchers. By inhibiting mTOR activation, rapamycin could potentially slow down the progression of senescence in the skin, halting the secretion of pro-inflammatory molecules that can cause damage to surrounding tissues that leads to visible signs of aging, like wrinkles, dyspigmentation, and skin thinning. In this study, published in the Journal of Geroscience, researchers set out to investigate the effects of applying topical rapamycin to the skin to see if it reduced the markers of senescence and aging in the human skin. The markers that were noted in the study were the P16 protein, solar elastosis, cytokeratins, and collagen production. The study was an exploratory, prospective, randomized trial that involved a total of 60 participants aged 40 and above. Participants enrolled in the study were provided with a container of rapamycin cream and a container of placebo in identical dispensers with labels for right or left hand, and were instructed to apply the creams to the dorsal side of each hand every 24 to 48 hours in the evening before bed. Before and after the treatment period, the researchers collected skin samples from the participants and analyzed them for markers of senescence and aging. They also assessed the participants' skin for signs of aging, such as wrinkles and sagging, using a standardized scoring system. The results of the study showed that topical rapamycin significantly reduced markers of senescence in the skin samples collected from the treatment group. The researchers evaluated the impact of rapamycin by analyzing skin biopsies for the expression of proteins related to senescence and aging. Shown here are the biopsies produced by this study. We'll go down the list of markers and explain how they relate to aging and cellular senescence. Now let's see how the hand treated with the placebo cream fared compared to the hand treated with topical rapamycin. The first marker is the protein P16. P16 is involved in the regulation of the cell cycle and inflammation. Because of its high levels in the skin, we can use it to track the severity and pervasiveness of cellular senescence and aging. in the rapamycin-treated biopsies, there was a 50% reduction in p16 expression relative to the control group. The fact that p16 was reduced in the rapamycin arm of the study suggests that the absolute number of senescent cells in the epidermis was reduced. The second marker was the condition known as solar elastasis. When we expose our skin to chronic sun exposure, we can develop a condition called solar elastasis. It is characterized by the accumulation of abnormal elastic fibers in the skin, which can lead to a thickened and yellowed appearance. Solar elastasis is often seen in areas of the skin that have been consistently exposed to the sun, such as the face, neck, and hands. When the researchers looked at the histological images, the placebo group had much higher levels of solar elastasis and disorganized collagen fibers that are associated with sun damage. The third marker concerns the distribution of cytokeratins in the skin. Cytokeratins are a family of proteins that are essential for the structural integrity and function of epithelial cells, including skin cells. In the study, the researchers observed that rapamycin improved the distribution of cytokeratins in the basal layer of the skin. This is significant because a well-distributed and properly functioning cytokeratin network is essential for maintaining skin health. The last marker pertains to collagen production in the rapamycin-treated group. Collagen, a structural protein found in the extracellular matrix of skin cells, is vital for maintaining skin elasticity and strength. Collagen 7 in particular is crucial for anchoring the epidermis to the underlying dermis and maintaining a functional skin barrier. As we age, collagen production decreases, leading to sagging, wrinkles, and other visible signs of aging. The researchers in this study found that collagen 7 protein levels were increased in the rapamycin-treated skin samples. Rather than simply modifying senescent cells present in the tissue, rapamycin treatment either reduces the number of cells entering senescence or increases the clearance of senescent cells. Whether the reduction in senescent cells is due to reduced entry into senescence or increased clearance, the reduction of senescent cells through the use of topical rapamycin improves functionality in the skin tissue. Beyond skin health, the implication of these results suggests that rapamycin could be a powerful tool to reduce senescence throughout the body. We now know about how mTOR and senescent cells drive aging, but what is the mechanism driving the opposite? Improving cell health, maintaining functionality, in some cases reversing damaged tissue, one dysfunctional cell at a time? We mentioned it briefly earlier, but the real cellular hero is autophagy. Remember how we talked about recycling cells? Autophagy is the cellular process that involves the degradation and recycling of damaged or unnecessary cellular components, such as misfolded proteins and damaged organelles. When topical rapamycin inhibits mTOR activity, autophagy increases and promotes cell health. Let's look at an example from before to see how autophagy improved and maintained tissue functionality with collagen production. Enhanced autophagy may facilitate the clearance of misfolded or damaged collagen proteins. By clearing out damaged collagen proteins, it makes way for the proper synthesis and assembly of new collagen molecules. This can lead to improved collagen quality and overall skin structure. This mechanism of pausing mTOR activation, slowing down senescent cells, and allowing autophagy to do its magic is at play in every one of the markers and in every cell. In conclusion, this study supports topical rapamycin as a potential skincare treatment, improving your skin not on a surface level, but at a cellular level. If you would like to continue receiving curated services, science updates, and news in the field of longevity, you can subscribe below, sign up to our newsletter, The Longevity Blueprint, or start receiving personalized longevity guidance today by one of our real doctors when you sign up with us today at gethealthspan.com.